my dear students, welcome back. In my last session we discussed Prophase 1. We completed Prophase 1. So continuation of that lecture, we will now continue with Metaphase 1. We will do with Metaphase 1. So at the end of Prophase 1, we find all the bivalents are scattered throughout the cytoplasm. And there is an increase in nuclear size and nucleolus. Now, in metaphase 1, the most important change which all of you know, you have studied in mitosis, is formation of spindle apparatus. You know, in plant cells, spindle apparatus is formed with the help of microtubules, and in case of animal cells, spindle apparatus is formed with the help of centrioles. So, a spindle apparatus is formed during metaphase 1. Now, these bivalents which are scattered throughout the cytoplasm, they are brought towards the center of spindle apparatus, which is called as equator. So, this is the spindle apparatus which is formed. You know, this is the equator. The center point is called as equator, and on the other side these are called as poles. These are called as poles. So all the bivalents which are scattered throughout the cytoplasm, they are now brought towards the center. They are arranged at the equator, forming an equatorial plate. Equatorial plate is formed by arrangement of chromosomes at the center. So all the chromosomes are now arranged at the center. Now how are they arranged? If we look at the bivalents, say this is one pair, this is one bivalent, this is another bivalent. So this is the centromere. So each chromosome of bivalent is attached to the poles. Each chromosome of a bivalent is attached to the poles. And you know, to the chromosome, to the kinetochore of chromosome, the spindle fiber is attached. So each chromosome of a bivalent, this is completely a bivalent. Each chromosome of bivalent is attached to the poles. And this process where all the bivalents are brought towards the equator, forming an equatorial plate, this process is called as congression. The most important change in metaphase 1 is called as congression. Congression means all the bivalents which are brought at the center. That is called as a congression. And number of bivalents, as earlier we discussed, depends upon the number of haploid chromosomes. And by this stage, the chromosomes become completely thick and rod-like. So, on the equatorial plate, we can find out the complete diploid set of chromosomes present at the equator. And what is the most important change is the congression. That is, all the bivalents are brought towards the center. And how are they arranged? Their centromeres are towards the poles. So, the important feature here is the centromeres are pointing towards the poles and arms are towards the equator. Arms are pointing towards the equator and centromeres are towards the poles. This is what is metaphase 1. Now let us see, compare it with next stage, that is called as anaphase 1. Anaphase 1. So the most important feature of anaphase 1 is separation of bivalents. Separation of bivalents. And this separation of bivalents, this is called as a disjunction. It is called as a disjunction or segregation. It is called as a disjunction or segregation. And here the chromosomes of each bivalent or each homologous pair it starts getting separated and it starts moving towards the poles. So the chromosomes now will start moving towards the poles. They are pulled with the help of the chromosomal fibers. Chromosomal fibers are those which connect the chromosomes to the poles. They are called as chromosomal fibers. You know, there are other types of fibers called as continuous fibers also, which run from one pole to the other pole. Continuous fibers are those which run from one pole to the other pole. Chromosomal fibers are connected to the poles. So, in anaphase, the chromosomes are pulled towards the poles and there is a separation of homologous pair of chromosomes. So, each pole will receive half the number of chromosomes. We started discussing the topic by taking an example of 2n equal to 4. So, in the daughter cells, we must get 2 chromosomes. So, this is the stage anaphase 1 where the reduction in chromosome number takes place. And each chromosome is having 2 chromatids. There is no chromatid separation here. There is only separation of the homologous pair without separation of chromatids. So, each chromosome will have 2 chromatids during anaphase 1. And this process is called as disjunction or segregation, and it is most important for, this stage is most important for reduction of chromosome number. Reduction of chromosome number. This is the most important feature of anaphase 1. Now let us go to the next stage, that is called as a telophase 1. Telophase 1. So I hope the points are clear and you have to note down the diagrams also as you are learning it, children. So now let us go to the last stage in karyokinesis here, called as telophase 1. Telophase, you know, are the reverse changes taking place to that of prophase. So here decondensation of chromosomes take place. Decondensation of chromosomes takes place and nuclear membrane and nucleolus reappear. And at the end of meiosis 1, we find 2 nuclei are formed. The 2 nuclei will have exactly half the chromosome number. That means they have 2 chromosomes each, and chromosomes again become thin thread-like and their identity again is lost. But each chromosome will have 2 chromatids. That means it has a 2 DNA molecules. And at the end of the topic, I'll give you a table with regard to chromosome number, chromatid number, DNA, and how are the changes in chromosome and DNA in different stages of meiosis. So here we find the chromosome number is reduced to half and each chromosome will have 2 chromatids. For the diagram purpose, I have shown 2 chromatids, but since it is completely decondensed state, the 2 chromatids are not very clearly visible. Now, at the end of telophase 1, cytokinesis may or may not occur. Generally, it may not occur. Cytokinesis occurs only at the end of second meiotic division. Now, let us see second meiotic. So, before I start with second meiotic division, we call the first meiotic division also as heterotypic division. We call it as heterotypic division. Meiosis 1 is called as heterotypic division, because at the end of meiosis 1, we find 2 nuclei are formed or 2 cells may be formed in any case. Then the 2 cells are not identical to each other. They have exactly half the chromosome number to that of the parent cell. So it is called as a heterotypic division, or this is the actual reductional division. Okay, now let us see the second meiotic division. Before the second meiotic division takes place, there is a resting phase between the two. A very short resting phase equal to interphase between meiosis 1 and meiosis 2. So, the stage between meiosis 1 and 2 is called as interkinesis. What is interkinesis? It is the stage between meiosis 1 and 2. And it is a very short stage. It is equal to interphase, but there is no S phase here. S phase or synthetic phase is absent. Only G1 and G2 phases are present. Only G1 and G2 phases are present, whereas S phase or DNA replication is absent. DNA replication is not required during meiosis 2. So therefore, there is no DNA replication, but only G1 and G2 phases. Cell cycle part again I will discuss in the next video. So cell cycle. So you know, but most of you know it. What are the changes in G1 and G2 phase and in the S phase? So S phase is absent here. That is the main feature of interkinesis. Okay. Now let us start with the meiosis 2. What are the changes or what happens in meiosis 2? So in meiosis 2, this is equal to mitosis. It is same as mitosis. Therefore, this is also called equational division. It is also called as equational division. It is same as mitosis. And at the end of this meiosis 2, 4 daughter cells are formed. From 2 daughter cells which are formed at the meiosis 1, 4 daughter cells are formed. And this also shows 4 stages like prophase, metaphase, anaphase and telophase. So it starts with prophase 2. So this prophase changes are same like mitosis. What are the changes in prophase? Decondensation of chromosome. Chromosomes remain as thin thread-like. And towards the end of the prophase, they become thick rod-like. And there is an initiation of spindle apparatus during prophase. So, the main features, condensation of chromosome starts and it's completed towards the end, and completely condensed chromosomes we find in metaphase. And prophase we also find there is after condensation of chromosome or after condensation starts. What is the most important feature of other features of prophase is initiation of spindle apparatus. Initiation of spindle apparatus. And lastly, nuclear membrane and nucleolus. Nuclear membrane and nucleolus disappear. Nuclear membrane and nucleolus disappear. These are the main changes. And since it is the continuation of meiosis 1, the chromosomes, each chromosome will have two chromatids. So chromosomes are with 2 chromatids. The number I am going to give you in my next video in a tabular form. Now let us see what happens in metaphase 2. Metaphase 2 is mainly characterized by formation of spindle apparatus and arrangement of all chromosomes at the center. So here two nuclei which are formed at the end of telophase 1, they are simultaneously participating in second meiotic division. So therefore two spindle apparati are formed here. So here we find two spindle apparati are formed, and these two spindle apparati are normally perpendicular to that of the spindle apparati formed during meiosis 1. So this is the equator. This is the equator. So spindle apparati formed here normally will be perpendicular. This is called as a spindle axis. From pole to pole, if we draw a line, that line that is called as a direction of spindle apparatus. So spindle apparatus in metaphase 2, we find they are perpendicular to that of metaphase 1. So here we have two chromosomes. We started with 4. So diploid number we are taken diploid number as 4. So here we find two chromosomes are arranged at the equator. So here you try to understand how the chromosomes alignment is taking place. How the chromosomes are aligned at the equator and how they are joined to the poles. How they are aligned and how they are joined to the poles. So here we find the centromeres of the chromosome, centromeres are at the equator. Arms are towards the poles, which was opposite during metaphase 1. Here centromeres are at the equator, arms are towards the poles. And each chromosome here also is having two chromatids. And you know each chromatid is equal to one DNA molecule. Each chromatid contains one DNA molecule. So two chromosomes are present at the equator. Very important point is centromeres are at the equator and arms are pointing towards the poles. The spindle fibers, the types of spindle fibers, all those are same. Like there are chromosomal fibers, continuous fibers and interzonal fibers also. Interzonal fibers will take part in the next stage, that is anaphase 2. Okay, now let us see the third stage, that is anaphase 2. So what happens? The main change in anaphase 2 is formation of daughter chromosomes or splitting of centromere. There is splitting of centromere, or there is a formation of daughter chromosome. Due to splitting of centromere, the daughter chromosomes are formed. Since centromere is splitting, each daughter chromosome will have only one chromatid. That means only one DNA molecule. So at each pole, if we observe here, at each pole, if we observe at each pole, we can find there is a splitting of centromere taking place. And as a result of splitting of centromere, separation of chromatids will take place. And these daughter chromosomes, which are called as chromatids, these daughter chromosomes will start moving towards the poles. These are the daughter chromosomes having only one chromatid. Having only one chromatid will start moving towards the poles. And in the mitosis, you must have studied that this is the stage responsible for different shapes of chromosomes. Depending upon the position of centromere, this shape helps in understanding different shapes of chromosomes. What are those different shapes? V, L, J and I shapes. So this stage helps in identifying these different shapes of chromosomes, that is V, L, J and I shape, depending upon position of centromere. So that is differently we can discuss in the structure of the chromosome. So the importance of anaphase 2 is formation of daughter chromosomes, and each pole is receiving two chromosomes. We started with 2n number 4. chromosome number is reduced to half. And each pole is now receiving 2 chromosomes, and each chromosome is having only one chromatid or one DNA molecule. Here each pole or at the center we have here 2 chromosomes, each one with 2 DNA molecules. So altogether we have 4 DNA molecules. Here also 2 chromosomes and we have 4 DNA molecules. But here each pole we have 2 chromosomes and only 2 DNA molecules. Each pole, because each pole is later developed into one daughter cell. That's why we are concentrating on each pole. Otherwise, suppose we think of the complete cell, because cytokinesis has not yet taken place. If we think of the complete cell, how many chromosomes we can find in complete cell? Each pole we are getting 2 chromosomes and 2 DNA. So complete cell, 4 poles. So we have total of 8 chromosomes and 8 DNA. So temporarily we can say in NF phase 2, the chromosome number will become 4 times. Each pole we are getting 2 chromosomes. So 4 poles we have 8 chromosomes, but we have 4 chromosomes here, doubling, not 4 times, sorry. Doubling of chromosome number takes place temporarily at this stage. Temporarily. But we know each pole is going to get 2 chromosomes and 2 DNA molecules. This type of analytical way of understanding will help you to solve many objective questions. In many entrance exams, based on the diagram or based on the concept, you are going to get different questions. In my another session, I am going to discuss the various questions from meiosis, and that will help you a lot. So I hope this point is clear. That is how we can see doubling of chromosome number. If we count the chromosome number at each pole, here we get 2 plus 2, 4. Here also 4. So total we are getting 8 chromosomes in that particular cell. But actually the chromosome number is 4. So temporarily, this word is very important. Temporarily, we find there is a doubling of chromosome number at this particular stage. So that is what is the NF phase 2. Now lastly, let us complete what is telophase 2. Telophase 2. So what happens in telophase 2? So telophase 2, the normal changes will take place. The routine changes of telophase 2, you know it. That is decondensation of chromosomes, and chromosomes become completely thin thread-like. There is a reappearance of nuclear membrane and nucleolus, and there is a formation of 4 nuclei. If cytokinesis has not taken place, then we find there is a formation of 4 nuclei. So cytokinesis now here will take place simultaneously. Cytokinesis will take place simultaneously. So 4 nuclei are formed, each one having 2 chromosomes. We find 2 chromosomes in each. So these 4 nuclei which are formed, each one will have 2 chromosomes. And now it is followed by cytokinesis. Cytokinesis simultaneously will take place. That means in both the directions. It can take place in this direction and also in this direction by formation of cell plate. So now let us discuss the process of cytokinesis. Little bit in detail I will discuss. So simultaneously here cytokinesis will take place. So it may take place at the end of meiosis 1 or it may not. That means cytokinesis point, if we take in meiosis, it may take place once or twice. Once means only at the end of meiosis 2. Twice means both at the end of meiosis 1 as well as at the end of meiosis 2. Okay, now lastly, let us see what is the process of cytokinesis. First, in case of plant cell, then I'll give you a simple way in animal cell. So cytokinesis. Cytokinesis, you know, it is a process of division of cytoplasm to form the cells. So here 4 cells are formed, and these 4 cells which are formed, they are not identical to the parents. Okay, this point again I'll give you in the significance also. So now let us see what is cytokinesis. So first step in cytokinesis is formation of phragmoplast. Phragmoplast is a barrel shaped structure. So suppose I'll take two nuclei, and between these two nuclei the cytokinesis has to take place. So first step is formation of a barrel shaped structure which is called as phragmoplast. This is a barrel shaped structure which is formed. This is formed with the help of spindle fibers. All these spindle fibers will break, they assemble in such a way to form this phragmoplast. It is a collection of all spindle fibers. Now, after formation of phragmoplast, the Golgi complex will secrete pectin. Golgi complex will secrete pectin into it, and now it is converted to cell plate. After Golgi complex secretes pectin, now when pectin gets deposited, then it is called as cell plate. And after cell plate formation, the cell plate will extend on both the sides. That is centrifugal growth of cell plate. Centrifugal growth of cell plate. Cell plate will grow centrifugally and then divide the cytoplasm. The next step in plants, this cell plate will be converted to middle lamella. Cell plate will be converted to middle lamella. By deposition of calcium and magnesium, the cell plate will be converted to middle lamella. So with this, the formation of cell plate is completed and cytokinesis is completed and divides the cell into 2 cells. So here we find in 2 directions simultaneously cell plate is formed, dividing the entire cytoplasm into 4 parts. And this type of cytokinesis in plants, it is called as a cell plate method. In animals we find much simpler method which is called as cell furrow method. This is called cell plate method, and in animal cells we find cell furrow method. There is no formation of cell wall, so it is called as a cell furrow method. Simply here what we find, there is no cell wall formation. So simply the plasma membrane which is present, this will show some invagination from one side, especially in the center it shows invagination. This invagination will grow up to the center, dividing the cytoplasm into 2. So it is called as a cell furrow method. So that is what is cytokinesis. So at the end of meiosis 2, when cytokinesis takes place, we find 4 cells are formed, and these 4 daughter cells are not similar to parent cell. They are not similar among themselves, and they are not also similar to parent cell. Now lastly let us complete last part of the topic here, that is significance. What is significance of meiosis? Significance of meiosis. Significance of meiosis. So in significance of meiosis, First important feature, it is involved in formation of gametes. Involved in formation of gametes. That is, formation of gametes is called as gametogenesis. Formation of gametes, or it is called as a gametogenesis. And there is crossing over and exchange of genetic material. Crossing over leads to exchange of genetic material. This exchange results in variations, which is very important for evolution. Variation we find between the parent generation and the progeny. Otherwise, the parent and the progeny would be exactly similar like carbon copies. That we don't find. We may find in some films, you must have seen. But we don't find that a father and son or a mother and daughter exactly look alike. Though there are some resemblances, but we find there are some differences also. These differences are mainly due to the crossing over which is taking place during Pachytele stage. And this variation leads to formation of new species. And it is very important for evolution process, and also it maintains constant chromosome number from one generation to the other. It maintains constant chromosome number from one generation to the other. Otherwise, suppose there is no meiosis occurring in any organism, especially sexually reproducing organism, then chromosome number will keep on doubling every generation. That leads to the death of the organism. Every organism has a particular number of chromosomes, and those chromosome number must be maintained. So therefore, it cannot go beyond that chromosome number. So this meiosis helps in maintaining constant chromosome number. Another last important feature of meiosis, it produces 4 daughter cells. Meiosis produces 4 daughter cells which are not identical to parent cell or among themselves. Not identical to parent cell or among themselves. So that means the daughter cells formed are qualitatively and quantitatively. They are different. Quality of chromosomes is different because of crossing over. Quantity of chromosome also is different because chromosome number is reduced to half. So here this point we can say there is a difference in daughter cells both qualitatively and quantitatively. That is very important feature of meiosis. So with this, children, I complete meiosis. In my next session, what I wanted to do is I have to give you a tabular column which help in understanding and solving many numericals related to meiosis. And I will also give you the difference between mitosis and meiosis. So these 2 aspects are left in meiosis, which I am going to complete in my next video. I hope you liked the video and you understood. More than liking, I should say, it is you have to understand the concept. It is not some entertainment channel where you like also, but of course you should like it. But more than that, you should see that you have understood. For understanding, you have to always watch any video from the beginning till the end. Then you will know whether you have followed or not. If you have understood my video, please subscribe and share to your maximum friends so that it will be more and more helpful to you and all your friends. So here I close my session, children. Thank you very much. Have a good day.